Friends, our today's video is about single base extension primers as well as SNP flanking or allele flanking primers designing. And we will use BatchPrimer3 server for this purpose. Okay, let's begin. Bismillahirrahmanirrahim. SNP or allele flanking primers means the same thing. However, single base extension or SBE primers are different from SNP flanking primers. I have already explained in detail about BatchPrimer3 server in my previous videos. Here in this video, I'm just going to tell you the reference of BatchPrimer3. The title of the article where it was published is BatchPrimer3, a high throughput web application for PCR and sequencing primer design. This article was published by these authors. This article was published in BMC Bioinformatics in 2008 on page 253. Now let's first see what SNV means. SNV means single nucleotide variations. And SNVs can be classified as synonymous SNVs and non-synonymous SNVs. The synonymous SNVs are those single nucleotide variations where the codon is mutated, but it still specifies the same amino acid. That is, it's a silent mutation. It doesn't affect the amino acid it codes for. So the DNA is mutated while the amino acid or the protein chain remains the same. On the other hand, non-synonymous SNVs are those SNVs in which the mutated codon either specifies a different amino acid or it doesn't code for any amino acid. That is, it's changed into a stop codon. For example, a wild type is specifying cysteine and the mutated one could be specifying glycine. So the amino acid it codes for is also changed. Those are non-synonymous SNVs. Then non-synonymous SNVs can be further classified in two categories: missense SNVs and nonsense SNVs. Missense SNVs means that the altered codon specifies a different amino acid. However, nonsense SNVs means that the altered codon is changed into a stop codon. It doesn't code for any amino acid. The missense SNVs are then further subcategorized into two classes, that is, a conservative substitution and a non-conservative substitution. A conservative substitution results in the replacement of one amino acid by another that is chemically similar. That is, although the amino acids are different from one another, but both the amino acids are chemically similar. So, the effect on the protein function is usually minimal. However, on the other hand, a non-conservative substitution results in the replacement of one amino acid by another that is chemically dissimilar, which often has a deleterious effect on protein function. Now let's understand each of these types of SNVs by examples. Let's suppose this is a gene for a specific protein and this is a wild type allele. As we all know that 3 nucleotides are considered as one codon and each codon codes for a specific amino acid. For example, here you can see that this AUG codes for methionine, which is also the start codon. And this second codon, that is UUU, codes for phenylalanine and so on. This UAA doesn't code for any amino acid. That is, it is a stop codon. So this gene, for example, is coding for this protein. So this is a wild type allele. Now let's see how synonymous mutation occurs or what synonymous mutation is. Let's take the same gene again and let's make a synonymous mutation in this gene. For example, we are changing this C. So remove this C and change this C with A. And now let's see what protein does it code for. The same. As you can see, this ACC coded for threonine and ACA also coded for the same amino acid, that is threonine. So both this wild type allele and this mutated allele, both are coding for the same amino acid. Although the codon has mutated, but the protein sequence was not affected by this mutation. So such kind of mutations are called silent mutations or synonymous mutations. Now let's take example of non-synonymous missense and conservative mutations. Let's take example of the same gene again. And let's now mutate this C with A. So drop this C and mutate it with A, or replace that C with A. Now this gene is mutated at this point. So this is a third allele of this gene. Now let's see what protein it codes for. As you can see, the amino acid that was coded by CCU was leucine, but now as the codon has changed, it is now coding for a new amino acid, which is isoleucine. But this new amino acid is chemically similar to leucine. That's why this mutation is called conservative mutation, but it's non-synonymous. Now let's take an example of non-synonymous missense but non-conservative. The non-conservative here means that the new code that is specifying a new amino acid will be chemically dissimilar as well. For example, here we are going to replace this U with G. And now we have changed this UGU to GGU. And let's see what amino acid it codes for now. As you can see here, UGU was coding for cysteine. But as we replaced this U with G, and now the codon is changed, and this codon codes for glycine. So these two amino acids are chemically dissimilar. So this mutation is called non-synonymous missense non-conservative mutation. Now let's see the example of non-synonymous and nonsense mutation, in which a codon is changed into such a codon that doesn't code for any amino acid. That is, the codon is changed into a stop codon. Suppose this is the same example of the same gene. So now here we are replacing this C with A. So replace this C with A. And now you can see that this codon has changed to a stop codon. Here this codon is coding for serine, but here when we replaced this C with A, it changed into a stop codon. So such mutations are called non-synonymous nonsense mutations. So these were the examples of all the types of SNVs. Now let's see what are single nucleotide polymorphisms or SNPs or SNPs. A single base sequence variation between individuals at a particular point in the genome is often referred to as single nucleotide polymorphism or SNP, and this is pronounced as SNP. SNPs are abundant in human genome and as such are being used for linkage studies to track genetic diseases. Millions of SNPs exist per individual, and thus the abundance of SNPs means that they could be used to help differentiate individuals from one another. SNP analysis is routinely used for linkage association studies, pharmacogenetics, and forensic, etc. SNP markers were categorized based on their applications. And SNP markers have been classified into 4 general uses. First of them is individual identification or identity testing SNPs. Lineage informative SNPs. The third is ancestry informative SNPs. The fourth type is phenotype informative SNPs. SNP or SNP, position in a genome where a person has a different nucleotide than the others at the same position. For example, this is the wild type allele. That is, most of the people of that population have this specific allele in their gene. But there might arise some people with a point mutation in this specific gene. For example, this is another person who has C in place of T in this specific gene. So it will create another allele with C instead of T. And this is its complementary strand. So it's clear that its complementary strand must have G, because C will always have G as a complementary nucleotide. So this kind of mutation will be found only in a few people of that population. So this is a SNP now. For example, there are some other peoples who have G instead of C in their positive strand. So this allele is now different from this allele as well as from the wild type allele. And there are 4 potential alleles possible for a single point mutation. As you can see, this could be T, it could be C, G, or A. So these are 4 possible alleles of the same gene. However, it's not always the case that you will find all these 4 different alleles of a single SNP in the population. Maybe you see only wild type and this allele in the whole population. Or maybe you only find this allele in the population. But I'm just talking about the potential. That is, a single SNP can have 4 different potential alleles. How can we identify SNPs? Although there are several methods for SNP identification, but in this video I'm going to tell you about 2 methods. One of them is called single base extension method and another is called SNP or SNP flanking or allele flanking method. So SNP flanking or allele flanking method is the same method. We will also talk about allele specific primers in my next video. Now first let's see what SBE means. It stands for single base extension. It's a method and it is used to determine the identity of a nucleotide base at a specific position in a genome. One such application is SNP detection. SBE primers are widely used in some high-throughput detection technology platforms such as snapshot applied biosystems and fluorescence polarization detection (FPTDI). This method was invented by Philip Gillett, Michael Knapp, Richard Douglas, and Stephen Anderson while working at the company Molecular Tool. This approach was designed for high-throughput SNP genotyping and was originally called as genetic bit analysis or GBA. How does SBE identify SNP or SNP? You will have to design a primer just one base before the SNP, either on the forward strand or on the reverse strand. As here you can see, we have 2 people with 2 different alleles. Here this person has G in its gene while this one has A in its gene instead of G. So these are 2 different alleles in 2 different peoples. So if you want to design SBE primers, then this is, as you can see, is SNP or SNP. So just before this SNP, design a primer which will be your forward primer. Or you can also design a primer, a reverse primer for its complementary strand. But that primer should be after this SNP, that is here. It should be adjacent to that SNP. Same is the case with the second person. So the same primer will be able to bind to this allele as well as to this allele because they are sequences. Why are they identical? Because we have not included this G in the primer as well as this A in the primer. So that's why they are identical primers. Now, after designing the primer, you will have to synthesize the unlabeled primers. After you synthesize those unlabeled primers, you will have to hybridize the primer to the target DNA. That is this DNA. This is your target DNA. So definitely it will bind next to the SNP on both the alleles. So it has the ability to bind to this allele as well as to this allele, and they will bind adjacent to the SNP. Now you will have to add 4 fluorescent terminator DDNTPs. Terminator DDNTPs mean that once they are bound here at the end of the primer, then there is no possibility that this strand will extend further. So you are using all the 4 terminator DDNTPs. So if any one of those 4 DDNTPs binds here, then this will stop extending further. One of these DDNTPs will extend the primer by only one base and then it will terminate there. No further extension is possible because all the NTPs are terminators. As all the DDNTPs are labeled differently, therefore the one that is bound can easily be identified. One such method is capillary electrophoresis fragment separation on the Genome Lab GEXP genetic analysis system. So how will you identify the DDNTP that is attached to the specific primer? Many approaches can be taken for determining the identity of a terminator. These methods include fluorescence labeling, mass labeling for mass spectrometry, measuring enzyme activity using a protein moiety, and isotope labeling. Mechanism for SBE. For example, to identify the following 4 alleles, we need to design a primer whose 3 terminal is attached to the base directly adjacent to the mutated nucleotide position. For example, there are 4 peoples with 4 different alleles. At this position, they have different nucleotides. As we already talked about 4 different possible alleles of a single gene. So this is the region of SNP. We have to design SBE primer for this location. Okay, now let's design SBE primer for these 4 alleles. Remember that primer must be designed in such a way that it binds adjacent to the SNP position. That is, for example, we designed this primer. Then this primer has the ability to bind all these 4 alleles because their nucleotide sequence is identical and it must not include this SNP position. That is, it should be adjacent. Or you can design primers that is on the reverse strand, on the complementary strand as well. For example, here, but the primers designed will be on the right side, that is on the 3 side of the strand. Again, they will not include this SNP position. So you design these primers. Now let me tell you about the procedure in detail. For example, to identify the following 4 alleles, we need to design a primer whose 3 terminal is attached to the base directly adjacent to the mutated nucleotide position. That is, these are the 4 different alleles and we have designed the same SBE primer for all of them. Now let's take 4 Eppendorf tubes, then prepare a master mixture in all of them and put each allele in different Eppendorf tube. For example, you put this allele in the first Eppendorf tube, the second allele in the second Eppendorf tube, this one into this third one, and this allele in the fourth Eppendorf tube. Now the second thing is the primer. So you should put the same primer in all these 4 Eppendorf tubes. After you put the SBE primers, the same SBE primer in all these 4 Eppendorf tubes, now you will have to add labeled DDNTPs. So all these labeled, that is 4 DDNTPs, will be added to the first, to the second, the third, and to the fourth. That is, to all of these Eppendorf tubes, you will have to add these 4 DDNTPs, which are already labeled. So what will happen next? As you can see that here in the strand of DNA, we have A next to T. So what nucleotide among these bind to this primer? Definitely T will bind here because we already know that this T is a DDNTP. That is, it is a terminator nucleotide and it cannot be extended further. So this will bind here. And what is the possibility here? We know that this is G, so only C can bind here. So C will come and will bind to this primer and will terminate this as well. The third will be terminated with G. So only G nucleotide will be added here. And the fourth will be terminated with A. As we know that all these 4 different nucleotides are labeled. So these labeled nucleotides can easily be identified. And we have also put all of them in separate Eppendorf tubes. And same is the case with the complementary strands, that is the reverse SBE primers. If you have designed reverse SBE primers, they will also extend by only one base. That is, here they will extend by A, Here G will be added and C and T terminator will be added to this fourth allele. Now let's compare the flanking and the SBE primers. So what does SNP flanking primer means or allele flanking primer means, and what does SBE primer mean? We already talked about SBE primers, those primers that are adjacent to the SNP position. But here you will see the SNP flanking primers as well. So the first example is for the SBE primers. This is forward SBE primer, this is reverse SBE primer. Here you can see that this primer and as well as this primer doesn't include the SNP position. That is, they are just adjacent to the SNP position. So such kind of primers are called SBE primers. Remember that in SBA method, you don't need both the primers. You can either use the forward primer or reverse primer. Either of those primers will work for you, depending on the properties of the primer. Now what about SNP or allele flanking primers? Here in SNP flanking, we need both forward and reverse primers. And the difference here is that the SNP flanking primers are away from the SNP position. The distance between SNP and the primer is not specific. So primers designed before and after SNP position that flank that region of the SNP position, they are called SNP flanking primers. So here we can say that they sandwich this SNP position along with nucleotides surrounding that SNP position. So the difference is clear between both the primers. Now let's use BatchPrimer3 to design SBE as well as SNP flanking primers. For designing SNP flanking or SBE primers through BatchPrimer3, you need to follow the following steps. The first step is that you need your sequence in FASTA format. The second step is that you must replace the nucleotide, that is ATCG, at the SNP position with other letters that shows nucleotide ambiguity. And those letters could be one of these, that is R, Y, S, W, K, M, B, V, T, or H, according to the following table. In this table, we have two columns. One represents the possible alleles of a gene, and in the next column, we have shown the replacement letter. For example, if there are two possible alleles of a gene, one with A and the other with G at a SNP position, then you can replace that A and G with R. If those alleles have either C or T, then you can replace them with Y. If there are two alleles, one with C and the other with G, then you can replace them with S. For A and T, you can make a replacement with W. For G and T, K is used. For A and C, we use M. If there is a possibility of three alleles, one with C, the other G, and the other with T, then you can use B. For A, C, and G, we use V. For A, G, T, you use D. And for A, C, and T, you can use H. But here you don't need to worry about it because you can use any of the replacement for any allele combinations. All of them will work the same way. But one thing is compulsory. That is, you must replace the regular nucleotide with one of these replacements. After you make replacement, then copy and paste the sequence in BatchPrimer3. The fourth step is that just click on Pick Primers. Your primers will be ready in a moment. So let's see. For example, this is a gene sequence and we have SNP at this position highlighted in yellow color, and one of the allele is with G and the other allele has T at this place. Then let's see the table and see what letter can we replace these two with for G and T. G and T can be replaced with K. So let's replace these two letters with K. The third step is that you need to copy this sequence. Go to BatchPrimer3, select SNP allele flanking primers, paste your modified sequence here, then just click on Pick Primers. Your forward and reverse SNP allele flanking primers are ready. This is your forward primer and this is your reverse SNP allele flanking primer. Same is the method for SBE primers. For SBE primers, you need to select this single base extension SBE primers. After you select this option, paste your sequence here. After pasting the sequence, just click on Pick Primers. Your forward and reverse SBE primers are ready. There is a third option here, which is called Single Base Extension SBE primers and SNP allele flanking primers. This option will design both kinds of primers, that is SBE as well as SNP flanking primers, in one go. Then just paste your sequence here, which is a modified sequence. We have replaced that G and T at the SNP position with K. And then you need to click Pick Primers. Okay, both the SNP allele flanking primers and SBE primers are ready in one go. For example, you have two alleles, one with G and another with A. Then you can replace this G and A with, let's see the table, G and A. This is G and A. We can replace this with R. So let's replace it with R. So now replace this with R. The next step is to copy the sequence. Go to BatchPrimer3. Select one of these three options: SNP, single base extension primer, and SNP and single base extension. But this is the best option because this will design both kinds of primers, that is SBE and SNP. Select this option, remove the previous sequence, paste your new sequence here, and then click Pick Primers. And in just a moment, it will make your primers ready. Okay, as I told you that you can replace the SNP position with any of those letters. As I just told you that this is not necessary that you must replace this AG allele with R. You can replace AG allele with Y, with S, with W, or with any other of these letters. So any one of these will work. So that was the method of designing SNP flanking as well as SBE primers using BatchPrimer3 server. So that was all about today's lesson, that is about SNP, allele flanking, and SBE primers. So thank you for watching my video. Bye.